Susana Fernandes, and I'm a chemist on biopolymers. And so my talk today, it will be divided in two parts. So first, I will describe a little bit about my research overview and interest, and also about my team. And then in the second part of the talk, I will describe some research achievements that we have that I have since 10 years now, working on marine bioresources. So, and regarding the research, I will describe a little bit what we are doing in the extraction of some biomolecules, and then the use of these biomolecules in the development of materials. So let's start by my research group and my research interest. So I work at the University of Po and Adour country. It's a small university in the southwest of France. It's very close to the border with Spain, and we have. So as you can see, we are a small university, but we have a very strong research areas. And for instance, we have 5 strategic topics like environment, energy and materials, bioresources, or even aquatic environments and resources, for instance. So I arrived here in France in 2018 to create a research group. So I'm chair on the valorization of marine bioresources and marine bio-inspired materials. So this research group is hosted in one of the research institutes of the university here, UPPA, and also the CNRS, is the National Center for Research in France. So it's in this research institute. So as I said, since now 3 years, I have created a group that calls MANTA. It comes from marine materials. And the aim of the research group is to develop bio-inspired and sustainable biomaterials to lower the impact on marine environment. Because as we know today, there are a lot of wastes and pollutants in the marine environment. So our aim is to develop new materials that will lower the impact of these materials. So to do it, we have 2 different approaches. So one is to valorize marine organisms wastes or byproducts, and as a source of new chemicals and new biomolecules. And the other one, to develop these materials, is to be inspired by some marine organisms, or even for some systems, marine systems. So we will try to find some inspiration in the functionality of these marine these marine organisms, or even in their structures and functionalities, as I said. So we have 4 main target applications for our materials. So biomedical, cosmetic, and also packaging, food packaging in special, or in particular, and also nanocomposites. So to do these materials, so we have 4 topics for 4 research lines in our group. So the first one is the valorization of, as I said, marine bioresources and fisheries byproducts. So we used, for instance, the skin of the fish, or the shell of some molluscs or crustacea, to extract biomolecules and biopolymers of interest. And then with these biomolecules and biopolymers, we do bio-inspired materials. So we can do, as I will show you some of these materials, we can do films, 3D scaffolds, hydrogels, composites, etc. But our aim, it's not only to produce these materials, but we want also to see what is the impact of these materials in the marine environment, as I said in the beginning. So we will study the impact of these materials on water, but also in some marine organisms, And we are also interested to see the impact of these materials in human health and in, for instance, in human cells, etc. So as I said, this is, so we have 4 different research topics in the group, and we start with 2 people, now we are 11 people. So, and we are distributed in these different topics. And recently we just we just hosted a new researcher. So we have a new junior chair, Manta Zymes. And the aim of this chair is to reinforce the needs that we have in biochemistry and enzymology. And Professor E will work with us in the 4 research topics that we have, but by helping us with this topic, biochemistry and enzymology. And also, we recently, we start work with other colleagues, for instance, economists or bioeconomists, and also some geographers, because we are also interested to know what is the future of our materials, or what are the materials of the future. So we have 2 main questions about it, about the development of these materials. It's not only the environmental impact, but also what is the impact, or what is, if we still continue developing this kind of materials that we are using marine bioresources. So we are interested to know about the bioethic or using these marine resources, and also about the acceptability of the society regarding these new materials, these new bio-based materials, in particular marine bio-based materials. So now I would like to move to the other part of this presentation, and I will present you. So first, how we are extracting some raw materials, so some biomolecules and molecules from these marine bioresources, and then I will talk a little about some materials. So, for instance, if we think about marine bioresources, as I said, what we are doing, we are trying to valorize some fishery byproducts and also some wastes. And we have 3 kinds of raw materials in this at this moment in the lab, for instance, from animals, plants, and algae. And we are trying to extract these or small biomolecules, so bioactive agents, or marine biopolymers by using green chemistry. So we are trying to develop different strategies or methods in order to avoid the use of organic solvents or high temperatures or the use of a lot of water. So regarding the materials that are the biopolymers that we can extract, for instance, we are trying to extract some alginate from brown algae, or hyaluronic acid, for instance, from the tuna eyes, or chitin and nanochitin from shrimp and crabs, and also collagen from the fish skin. As I said, we are also interested in small molecules. And for instance, here in France, in the Basque country, where we are, we have a lot of red algae. And we are also trying to valorize other biocompounds of red algae, because here it's the main compound that we are valorizing is the agar, and agarose. But we know that the red algae, they also have other molecules of interest. So like minor compounds like mycosporins or flavonoids, or some proteins. So in this case, for this presentation, I was choosing the chitin, so the valorization of the chitin. So what is chitin? Chitin is the main compound of the crustacea exoskeleton. So like cellulose is a linear polysaccharide and has a structural function in the exoskeleton of the shrimps and crabs. And as you can see here, the only difference with cellulose is in the carbon 2, because instead to have an hydroxyl group has an acetyl group. So it's very important, or a very interesting biopolymer for different applications, because it's biodegradable and is biocompatible. And as I will show you, chitin also show antimicrobial properties. But as you can see by their structure, chitin is or have been considerable an intractable polymer. Why? Because it's very hard to dissolve chitin, and this limits their use to do materials. So since long time, the main derivative of chitin is chitosan. So if we do some deacetylation here, we can have amino groups. And in this case, we have what we call the most important derivative of chitin, that is chitosan, that is soluble and that is also biodegradable, biocompatible, and presents antimicrobial properties, and very important, has film forming ability. So we can easily to do a solution of this polymer and then to transform it in a material as we wish. Fibers, transparent films, hydrogels, 3D scaffolds, etc. So this, it was until, I don't know, maybe 25 years ago, the main, and still the main derivative of chitin. But today we know a new one, a very interesting new derivative of chitin, that are chitin nanoforms. Because as I said, so chitin in the exoskeleton of the crustacea and also insects. So what happens is that the chitin molecules, they assemble together, and then we can have what we call chitin nanofibers. And then we have together, these nanofibers will be assembled with proteins and with minerals and do the composite that we have here, that it will be the crustacea exoskeleton. So what is interesting is, like in cellulose, we can take the crustacean exoskeleton, we can remove or extract chitin, and then we can extract chitin, nanochitin. So if we used chemical treatments by acid hydrolysis, we can have what we call chitin nanocrystals. And if we do some physical treatments or mechanical treatments like grinding, microwaves, and high pressure homogenizing, so in this case, we can also have what we call chitin nanofibers. So in our lab, we have developed two methods. So this work, it was done when I was working in the Basque Country University in Spain. So we have developed for the first time or extracted for the first time chitin nanofibers, as you can see here by these microscope pictures. These microfibers are very long, and we did it by using the high pressure homogenizing. So what was important here is that we have these, as I said, long nanofibers with a very, very high aspect ratio. And we were also interested to compare so the antifungal activity of these nanoforms, of these nanofiber forms with chitin nanoforms. and also for sure with our control material that was chitin. And we observed that the nano morphology of chitin nanocrystals or nanofibers, they presented better antifungal activity than the chitin. We see here that there are a little differences between the material or the chitin nanocrystals and nanofibers. And we think that is for two things. One is that in the chitin nanofibers, maybe the amino groups, because we have some amino groups in the nanofibers, are more available. And also because in this case, the degree of deacetylation, it was higher than for the chitin nanocrystals. So another method that we have developed to extract chitin nanoforms, it was microwave-assisted extraction. It was also work that we have done in collaboration with the Basque Country University. It was the work of a PhD student, Prof. Fernandes. It's a work that has been published this year. And we tried to extract chitin from 3 different raw materials. So squid, pen, shrimp, and lobsters. And we used the box experimental design and the response surface methodology to see what were the best conditions. So we work with independent variables. So we were interested to see the influence of the concentration of the acid that we used for the isolation of nanocrystals. We were also interested to know the time and the power. And we were able to have very high isolation yields. And the most important is that with the microwave-assisted extraction, we were able to reduce the time to 10-30 minutes, depends of the raw material. For instance, we were able to isolate in 10 minutes for shrimp. So for alpha chitin, but for beta chitin, we need more time. It was around 30 minutes. But what is most interesting is that with the conventional way, with an acid hydrolysis using conventional ways, usually this isolation takes around 90 minutes. So we were able to decrease the time and also the concentration of the acid. So the question that we were trying to answer, it was why to use these nanocrystals for to do some composites, and we were also interested to know the difference between use nanocrystals and nanofibers. So we did a lot of composites using different biopolymer matrices, elaborating by different processes, so casting, melting, film molding, freeze drying techniques, and then for sure we characterize all of these materials. So here, for instance, I have a comparison. So using starch as matrix, so starch is also a biopolymer, and we were trying to see what were so the differences between to use these two methods and the two kind of morphology, so nanochitin and nanofibers. So starting by the casting evaporation methods that we used to prepare films. So we were also interested to see the antifungal activity. And we observed that when we incorporate different amounts of chitin nanocrystals or nanofibers for both morphologies, we observed that an increase very considerable, so around 90% of the fungal growth. And regarding the mechanical properties, again using casting evaporation, so we also observed that the materials prepared with chitin nanofibers showed better mechanical properties than the materials prepared with chitin nanocrystals. We proposed that this is because of the percolation effect of these long nanofibers. So if we compared also the mechanical properties, but using another process, so the melting mixing process, we observed exactly the same thing. So with the two methods, casting or melting mixing, we observed the same thing. It's the incorporation of the chitin nanofibers that show the best mechanical properties. So what is important for these two kind of works is that the final properties can be controlled by the nature and the amount of chitin nano forms that we can incorporate in this biopolymer, but using different ways to produce it. So another thing that we were interested, it was mimicking the extracellular matrix to design biomaterials for cartilage. So in this case, we're still using chitosan, the two chitin nanoforms, so nanofibers and nanocrystals, but we changed the matrix. So now we are using chitosan as matrix. So we prepared two kinds of materials. So transparent films using, again, casting evaporation method. And we used freeze drying to prepare 3D scaffolds. So here we also see what were the best mechanical properties. And here we observed that for both materials, we observed that here it was the chitin nanocrystals with chitosan that showed the better mechanical properties. Maybe because the fibers, the chitin nanofibers, we were very long and we were not able to disperse very well in the chitosan matrix. So we showed that both materials using both morphologies, they show very good cell viability. We can see here the SEM images. So these are the aspects of the materials, the surface of the films and the porous scaffolds. And here in green, we can see the cells that are adherent to the material and proliferating. So then we used the materials that presented best conditions to prepare to use this material to do some differentiation. So we want to do cartilage. So what we did is that in this case, we used stem cells in the form of steroids. And we used the scaffolds or the 3D porous scaffolds prepared with chitin nanocrystals. And we were very happy because we were shown that we were able to differentiate the stem cells. And we also observed that these 3D porous scaffolds allowed the production of chondral extracellular matrix with significant sulfated glucosaminoglycan synthesis. So just to finish or before, so yes, these are some reviews that we were invited to do about chitin nanocrystals works. And now just before to finish my presentation, I would like to show you another material that we developed that is inspired by the mucus of the reef fish. And we tried to prepare some materials that absorbing the UV radiation. So it's very interesting that there are some algae and cyanobacteria, but here we are interested in algae, for instance, the red algae. They biosynthesized small molecules that are able to absorb the UV radiation to protect themselves from the UV radiation. So the fish, they are like us. They can't produce or biosynthesize these biomolecules, so they eat this algae, and then these small molecules, they appear in their mucus and also in their eyes, in their lens. So these molecules are secondary metabolites that we know two families, the mycosporins, like amino acids, and mycosporins. And what is interesting is that they absorb in the UVB and UVA. So it could be a good alternative for the use of synthetic UV filters or even nanoparticles. And it's very interesting because we have different functional groups that we can use it to graft these molecules to our biopolymers. And they have a great capacity of absorption, and they also show it antioxidant properties, etc. So we try to use these molecules to modify chitosan and try to do some materials. Here we can see this transparent film, and we can show that we can have some absorption in the UV region. And here we have shown by using murine fibroblasts that these materials also present good, they are not cytotoxic. So this work, we still working on it, using other biopolymers as a matrix to prepare this kind of very interesting materials. So just before to finish, I would like to thank to the people that are working now in my group and all the institutions where I worked before to be here in France, and also the funding projects. Thank you very much for your attention, and please, if you have questions or if you would like to collaborate, I'm here. Thank you.